Thank you, Ma'am Jessica. Again, hello everyone. I am Teacher Ginis Dacayo and I am going to discuss with you the ex vivo gene therapy. So the term ex vivo is associated with various biological processes that are made to occur in or on a biological tissue outside the organism. Etymologically, ex vivo is a Latin word which means from life. Ex vivo is also term used to describe a biological process that is carried out within or on a biological tissue in an artificial environment, but with the minimum alteration of natural conditions. In ex vivo gene therapy, genes are transferred to the cell grown in culture. Transformed cells are selected, multiplied, and then introduced into the patient. The use of autologous cell avoid immune system rejection of the introduced cell. When we say autologous, it is designed to use a person's own blood stem cell and insert into those cells a working copy of the missing or faulty gene. The cells are sourced initially from the patient to be treated and grown in culture before being reintroduced into the same individual. This approach can be applied to the tissues like hematopoietic cell and skin cell, which can be removed from the body, genetically corrected outside the body, and reintroduced into the patient body where they become engrafted and survive for a long period of time. For better understanding, here's an schematic diagram for ex vivo gene transfer. The first process is to harvest all the cell together with the cell with defective gene from patient. The second process is to transfer the cell to make them grow in culture, let them multiply, and then introduce into the patient. The third process is through the use of autologous cell to avoid immune system rejection because it is the time to combine the artificial RNA to the normal gene. The fourth process is the reverse transcription in which an enzyme makes a copy of DNA from RNA. The fifth process is the translation of the correct protein in cytosol because the translation of all proteins in a eukaryotic cell begin in the cytosol except for a few proteins made in mitochondria and chloroplast. The sixth process is to transfer all the transfected cell, or in other words, all those recovered cell. The seventh or the last process is to select, grow, and test all the transfected cell or recovered cell, grow cell to 100 billion to make the process of reinfusion of engineered cell to the patient. That's all about ex vivo gene therapy. Again, this is Teacher Ginis Dacayo. Thank you for listening.